Hello, in this video we are going to cover the enrichment analysis within GENAVi. I'm running a local version of the app, and simply this enrichment analysis comes after the differential expression analysis. From the DEA analysis, we're going to have the DA62 analysis, which results with simply this table, where we have the log two-fold change and the p-value adjusted. From this, you can identify which are the upregulated genes and downregulated genes. And now your question is, okay, what are those genes doing, what they're related to? And for that, we do a enrichment analysis. So basically, before I start showing the GUI, just to cover a little bit the background, if you go to Help Material, you have Learn More. You're going to arrive to this page here, which is called Pathway Analysis Workshop from Professor Gwen Guichon Yu. So he is the major creator of the packets that are using for the enrichment analysis, and it's covered in this workshop. So in case you use the GENAVi tab in enrichment analysis, please cite his package. So first, just to define, gene sets are actually lists of functional related genes, but there is no specification of relationship between them, while pathways are specific gene sets, represent a group of genes that work together in biological process. So imagine you have like cell cycle genes. So you have a list of genes that are working that biological process. So the annotations available in several databases such as Gene Ontology, Kyoto Encyclopedia of Genes and Genomes, molecular signature databases, disease ontology, disease network, and wiki pathways. So each database is going to have some kind of annotation. So for example, the MSig database is going to have the hallmark set genes, immunology gene sets, and etc. So basically using those annotations, one, okay, are my upregulated genes enriched, overrepresented within those annotated genes? And to evaluate that, we have two types of analysis. One's called over-representation analysis, ORA, and it's used to determine whether non-biological functions or processes are overrepresented, such as enriched, in a gene list. So basically, when it's to say if your differential expressed genes are enriched or not. So basically, what they simply do, just an example, would be like a Fisher test. So imagine you have 25 genes that are upregulated, and they are within that pathway, but 127 that are upregulated are not. Compared to all genes, the ones that are not upregulated, 237 are within that pathway, while at 11,000 are not. So if you do the ratio here, you can simply see there is enrichment, but you can use like a Fisher test for that, and then you can see it's significant and how big is our enrichment. And the second type of analysis is the gene set enrichment analysis. So the problem of the ORA is that you need to define a list of genes based on the threshold. So if you change the threshold, you have a different list. What if I could rank all genes based on the p-value or the log-fold change? And that's what the GC does. It's going to rank the list, the genes. So your input now is not a list of genes, it's the ranked gene list. And then you look for if a set S is enriched within that list. Either the top of the bottom or randomly distributed, which would be that set is not enriched on the top of the bottom. So if the list is ranked by log two-fold change, imagine that my top of the list is going to be the ones that are mostly positive and the ones at the bottom most negative, so downregulated, upregulated. And then just check if the S is within the up or downregulated. Then we're going to have an enrichment score and a significance level. And then the rest of this workshop is just giving examples how you can gather data and perform data analysis and plots. And the code we have here is exactly the one we are using GENAVi. Okay, so going back to the GUI, the first step is to upload the results. So I'm going to upload that table that we showed that has the breast cancer subset analysis, the luminal versus basal. And then you perform the analysis, okay? So we're going to perform the oral analysis, then you can select the pathways. And the second part here is just the cutoffs to select the upregulated genes or downregulated genes. Some databases are bigger than the other ones. So KEGG analysis, you can see, is taking a little bit more to execute. We have the plots and we have the tables, okay, which you can later read a little bit in the workshop tutorial and have a better sense of what they mean. In terms of the GUI, you can change the plots, like either, at least for the arrow, have the network or the dot plot. Then you can select the, okay, I just want the top 3 categories. You can export the image as PDF or PNG or SVG. You just need to change here. That's a high resolution one. Okay. And then one thing you can do is generate an HTML report, which is going to contain both the code and the figures used in the analysis. And then you have all the steps you need to perform the analysis. So we're going to show the data we loaded, which is the same as the table. We can see we're using gene symbol here, but all those pathways are annotated using gene and trace ID. So we need to use biomark and map those symbols to trace ID and sample ID. The sample ID is used mainly for the Go analysis. The other database are still using trace gene ID. And then the next steps, like how you perform the analysis. So we select the upregulated genes, those are our cutoffs. Then you select from the DA results the interest ID genes from the ones that are significant based on our cutoffs. So we have like 2,000 genes that are upregulated. Then you do then compare to the background genes, so 23,000. We're comparing using the KEGG analysis, and then you have the functions and the code that you can perform the analysis and plots. So you can easily change those by hand later if you want. Okay. So just to show if you perform a JC, I'm going to select another database, let's say like this. So JC, now you have the option to select actually the ranking method instead of the cutoffs for the different expression genes. Then you might have the same things, both plots and tables. But plots, now we have a different ones. We have the ridge line, you have the running score, draw linked list, ranked list, and enrichment network. So they are described in the workshop. I'm not going to cover in this video right now the meaning of each one. So that's how GENAVi is working. Please don't forget to cite, if you use this tab in the app, the packets from GENAVi on you. It's quite important not to forget about that. Thank you very much for watching the video. I hope you like it.